Welcome everybody to this week's edition of the EV Journal Club. We are coming to you today with a presentation from the west coast of the US. So we're welcoming Randy Carney from the University of California, Davis. And he's going to be telling us about nanoplasmonics. So some of you may be familiar with this field, but many of you probably are not. And so I think this is going to be a very educational talk for us today, and we'll open our eyes to some newer possibilities in EV characterization. So Randy, thanks so much for joining us today, and I'm going to hand this over to you. What we'll do here is ask everybody to please keep themselves muted during the presentation, and to use the chat box for any comments and questions that you might have. And then at the end of the presentation, we can open this up for discussion. So thank you all for joining. And Randy, it's all yours.
Great, thanks a lot. Welcome everyone, and especially thanks to Ken and his group for this, really nice invite. I think it's great to have this forum to chat about EVs. Clearly one of the good things that have come out of this global pandemic. And I, for one, hope that you guys are able to continue this for a long time, even after things may get back to normal. I think it's really cool. Okay, so let's jump right in. My goals for these next, 30 to 40 minutes are pretty simple. I want to break down very briefly what are the kind of important aspects of these light matter interactions that lead to these crazy, weird plasmons that I'll introduce. And throughout this introduction, which is pretty focused on kind of the basics of this, because I understand many of you may not be familiar with this type of physics, I'll sprinkle in some of the most current and cool studies that are utilizing these methods to detect and characterize EVs. So then I'll spend the very last part of the presentation sharing a sort of real example that's in progress now in my group. It's actually under review. We got a request to do more experiments, which is challenging at this time, as I'm sure you guys, many of you are in that same boat. And you'll sort of see that we're using these new physical materials to get at the same questions that everyone else is studying in EVs. And that is, what are the features of the heterogeneity of EVs chemically that we can exploit for diagnostic use. Okay, so based on my participation in this journal club the past few months and in the EV field in general, I'm guessing many of you don't have a kind of direct background in physics or material science, and which is, by the way, how I feel when I'm regularly Googling what the difference is between RNA and DNA. Probably most of your children have a better understanding of physiology than me. So it's nice to be on the flip side today. But in any case, I kind of want to start really simple to consider what happens when light interacts with matter, period, which is really the origin of any photonics effect, from fluorescence to light scattering, which are very commonly used in our field, but especially for plasmonics that I'll build up to. And so the study of light and matter interactions is called spectroscopy. And as depicted in this cartoon here, light interacts with matter in a bunch of different ways. It can be transmitted totally through, could get totally absorbed, reflected or scattering, and any combination of these. And so when you're looking at light coming from a material, you actually don't know necessarily, without careful experimentation, what the photon has done in interacting with a particle or an EV or material, whatever material. So it's important to know that both the material and the color, so the wavelength or the frequency, I'll use those all kind of interchangeably, affects this interaction. So different colors might reflect or might scatter preferentially. And a substance, for example, could appear blue if it absorbs all the red parts of the spectrum. It might appear blue to us if it scatters blue. So there's this important difference. And some materials you may see no light at all if it's completely absorbed, and these would appear black. And I kind of put the part of the electromagnetic spectrum up top here, 400 to 750. That's what we see with our eyes. Now there's lots of other materials that can do all of the effects I'm going to talk about today outside of the UV vis and infrared region. They're not as useful for us for diagnostics. It's much easier to detect the UV vis colors. Okay, so when nanoparticles of any type absorb light, the observer actually sees the light that's transmitted through the formulation. So we actually perceive the complementary color to the absorbed color. So this is the absorption spectrum for small gold nanoparticles shown here. And you can see that these are mostly absorbing in the blue and green regions of the visible spectrum. So if we were to shine sort of white light at these particles, the blue and green light is preferentially absorbed and transformed mostly to heat. And then the red light is transmitted through the material. So by eye, this would actually appear to be red, as shown here in that picture. But for scattering, it's the opposite. You actually are seeing the apparent scattered light color, the true color. And we typically collect it off to the side because scattering is happening it's shooting out at every angle, it's random. And so if we want to differentiate that from the transmitted light, we pick it off at a different angle. And this is how flow cytometers work and lots of different NTAs and all of these different machines that use the scattered light that we're familiar with. So these are some silver nanoparticles that are preferentially scattering blue and green light, as you can see in this dark field image. So basically what we can say is that the color of the absorbing material is everything not absorbed, and this color of the scattering is everything that is scattered. So it's kind of interesting. And because some materials are better at scattering or absorbing in different areas of the spectrum, gold versus silver, different sizes, we'll get into that shortly, you can actually mix a lot of these together to get cool dichroic effects. So many of you might be familiar with the Lycurgus cup. It's this Roman artifact circa 300 AD. And they unintentionally, well, they didn't know they were nanoparticles, but they certainly intentionally were making cool color materials. These nanoparticles formed during the glassmaking process. And you can see that the scattered light is dominated by silver particles, and you see this greenish tint looking from any angle. But if you transmit light through it, you can actually see this red color because the gold nanoparticles are absorbing the green and blue. So really the takeaway point of this introduction is just that we can actually engineer scattering and absorption into materials. And these so-called plasmonic particles are ones that can both absorb and scatter light. So they have dual functions. So the final piece before getting into how we can exploit this for chemical sensing for EVs, for example, is to understand actually how broadly and how finely we can modify optical properties just by making very subtle changes in size and shape of nanoparticles. So here's a UV vis spectrum, and it's just like you would get in a typical plate reader if you're doing VCA assay or something. It's exactly the same. And we can see the absorbance of these panel of hollow gold nanospheres, which you can make with pretty straightforward chemistry in a hood. So these are basically gold shells. So if you can see the sizes in this inset here, these are ranging from just 25 to 45 nanometer in diameter, very small change compared to what we're used to looking at for EVs, or 2 to 3 nanometer differences in the thickness of these shells, and we can sweep basically their absorbance color over the entire visible and near infrared spectrum. And for gold nanorods, you can do something similar. You can increase the aspect ratio, make them longer, and you can play with the temperature and different chemical properties while you're synthesizing these to do this. And you can, again, shift the peak absorbance. So keeping in mind that this absorbance generates local heating, as let's say you want to do thermal ablation therapy, you can insert these into the body and focus light right to the tumor and destroy the tissue. But let's say you only can apply light at 600 nanometers, that has a very thin penetration depth through the skin, gets attenuated too quickly, not much use. But if you make the particles longer, you can increase this resonance wavelength. So now you can have deeper penetration for more effective therapy. So there's a real practical use for these. And we just have great control over all kinds of particle shapes and sizes and grain structures and composition, their solubility in different media by functionalizing their surfaces with different molecules, attaching antibodies and peptides and other affinity agents. So the name of the game really is optical tuning to reach your desired application. So one of the probably most famous and awesome applications of plasmonics for sensing is this paper here from Nature BME a couple of years ago. And they're measuring EVs, as you can see. So they have antibody microarrays used to capture EVs. They had anti-CD81 here, and then complementary antibodies that were functionalized to gold spheres, these green particles, or gold rods, the red ones. Those had anti-CD63 and CD9, so your classic tetraspan panel. And these different shapes have different colors, like I was just showing you. And so because of these different properties of the rods versus the spheres, you can actually see that the apparent color when collecting the light using a spectrometer and CCD camera, very, very simple equipment, you can actually tell these two apart. But as you can see in the bottom right full color image here, or in the top left spectrum, If both particles are present in the same subdiffracted spot, in other words, they're co-localized, let's say on a single EV, you can actually see this intermediate yellow spectrum, color mixing. So one really key thing that's important to note here is that using these particles that are very small and very bright, you actually can beat the diffraction limit that plagues most optical techniques. So because of the diffraction of light around objects, there's essentially a fundamental physical limit called the diffraction limit that we've known about for hundreds of years. And it basically prevents us from resolving any features of objects that are much smaller than the wavelength of light that you're interrogating with. So practically for direct imaging using any visible or infrared light, you can't resolve objects less than about half their wavelength on average. And that's if you have very nice equipment. So practically speaking, it's usually higher than that. And of course, all small EVs that we're interested in fall well below that limit in the UV vis. And so it's basically this fundamental restriction of why we can't, for example, use fluorescence to image single EVs uptaking into cells. Now, of course, there are new super resolution approaches, but these are quite difficult in practice, very expensive, require a ton of expertise and training. And they're actually statistical approaches primarily, and you use to localize events smaller than the diffraction limited spot. But there are other tricks to get below this diffraction limit, and this is a field called sub-diffraction imaging if you're interested. And using the apparent scattering from these small nanomaterials is one of the primary methods to do this. So we can localize them smaller because of how bright they are. And it's kind of an averaging technique. So in this case, in the simplest form, these plasmonic particles can just be used as labels, though admittedly, you can't get that many colors out of them. So in this, even though they're much brighter than fluorophores, a lot more robust, they don't blink, they are larger, so they're bulkier, that's a problem. You have multiple antibodies probably, so there's an avidity issue. But you can sort of think of them as contrast agents or probes that you can multiplex to about the same degree as fluorophores. So here's an animation of actually how you can image scattered light. You might be wondering how they can actually obtain those. So in your optical path, you put this little disc that essentially pushes your light outside of focusing on your sensor. But if you're able to put your sample in the light path, you can see something coming in here that scatters the light. Some of that scattered light can be focused and collected on the detector. So this is called dark field microscopy. And it's a nifty way to directly image scattering in line with your transmitted light. So that's how that study was working. And they moved past the tetraspanin panel eventually and started looking at this EPHA2, FA2 expression. It's a pancreatic cancer marker. And they showed that in EVs from mice plasma, they could actually monitor tumor progression with this plasmonic labeling approach, as you can see in the top right here, just by looking at these raw data, these dark field images, and calculating the apparent colors. So they showed that they could use 150 times less plasma compared to ELISA. They can get down to 1 microliter of biofluid. They didn't have to isolate past the initial 10,000 G spin, and they showed pretty convincing sensitivity and reproducibility. So it's pretty exciting. Okay, so another way to sort of say that these particles are very good scatterers is to say that they're extremely refractive. So their refractive index, or how much light is bent upon passing through, is very, very, very high, much higher than organic materials. So scattering plays a huge role in many of our core instruments used to analyze EVs, like nanoparticle tracking analysis, NTA, and flow cytometry. And that's where these individual particles are all detected based solely on the detection of the light they scatter into a detector. So many of you may be familiar with this simulated data on the left from Josh Welsh and Jennifer Jones at NIH, and Edwin Vanderpool and his team are also doing very similar things. And it's sort of showing the scattering of 488 nanometer light as a function of diameter for low refractive index materials like EVs or polymer beads that are often used as reference particles. And because of their really low refractive index, it's extremely hard to actually collect enough light from vesicles less than 100 nanometer, and impossible to collect light for ones less than about 70 nanometer. And this really has led to this classic artifact in the EV field, where EVs have been reported to have this mode in diameter around 120 nanometer, as you can see here in the red and the NTA graph on the right. But this just isn't true. Many orthogonal non-light scattering methods of EV sizing that are capable of seeing much smaller aren't limited by refractive index, for example. They show that the log concentration increases linearly down to 50 nanometer and even smaller. So by the way, this is called this kind of log linear with size distribution. It's called a power law distribution, and it's a hallmark of aggregating systems in material science. And basically, there's absolutely no reason to expect that EV should magically stop assembling at some preferential diameter of 100 nanometer. And actually, interferometry is another way to beat the diffraction limit. You interfere a reference beam of light with the beam of light that's passed through the sample after it's passed through, and you can measure subtle phase shifts in the beam. You can size down in principle to even single molecules, and that's how the new NanoView ExoView instrument works. And if you're interested to learn more about that, I gave a talk with NanoView a few weeks ago that's available on YouTube with this QR code here. And I showed a bunch of our initial data with this instrument. I know Ken's lab is working with this one too. That talk is more focused on the current methodologies used to analyze single EVs and what are the trade-offs you might have in using them. So feel free to check that out if you're interested. Okay, so back to this high refractive index of these metallic nanoparticles. Continuing in this study, Josh at NIH, basically, they're experts in single particle flow cytometry. They showed the potential use of these high refractive index materials as scattering probes for flow cytometry. So in this data on the right, you can see various sizes of silver and gold nanoparticles in red and blue. They're increasing in increments of 20 nanometer in size, and they're being picked up, this is real data, by the Beckman Astrios flow sorter. And then you can see that compared to these polystyrene particles in green, which even have a higher refractive index than EVs, so you would even see larger EVs at this size, the sensitivity and size is much less. So these metal nanoparticles have refractive indices that are nearly twice that of EVs. So you can see them at effectively much smaller sizes. So this is important because the size limit that we run into with EVs on many of these instruments, it's not directly caused by their diameter per se, but indirectly it's actually caused by their refractive index. So if you can load your EVs with gold particles or on the surface or on the inside, you could boost a lot of these inherent scattering issues. So in my lab, for example, we regularly use NTA to analyze gold particles down to 20 nanometer. They're so refractive, they can be viewed extremely easily. So at this point, hopefully you're pretty comfortable with the fact that these nanoparticles of various sizes and shapes have specific interactions with light that allow them to scatter, absorbed, or refract light at different wavelengths. And in and of itself, they're very useful as sort of these colorimetric contrast agents for EVs, sort of just like ultra bright, ultra refractive labels. But you may be wondering, how do they actually do this? What is the physical nature of these metal particles that enables them to scatter such high light? And that is the plasmon, this funny named little physics wave. So as it turns out, the sort of remarkable optical properties of what I'll now call these plasmonic materials, and they're mostly metals, they occur upon the coupling of the incident photons from the light with the electrons in the material. The electrons are actually the key. So the electrons in metal are special. They're much different compared to typical organic material that you would find in a lipid, for example. So in metals, there's so many nuclei, let's say gold atoms, packed together in a tight crystal lattice that all of their valence electrons, these are the ones in the outermost energy shell, if you recall back to your chemistry studies, those free electrons move around the material like a plasma, hence the name plasmon. They're no longer confined to the atom from which they originated. And this is called delocalization. They're delocalized from their original location. And it's what gives rise to the electrical conductivity of metals and also this strange interaction with light. So these free plasma-like conduction electrons, in response to certain frequencies of light, certain wavelengths, undergo an actual collective oscillation. And that's what's sort of pictured here. The electrons are resonating with the incident light radiation. So at any given time, there's a depletion of electrons at one side and an excess of electrons at the other side. And they're oscillating back and forth as long as the light is on. Wah, wah, wah. And so basically, this is called a localized surface plasmon resonance. And it results practically, and this all comes out in crazy physics and even quantum physics if you really want to understand what's going on. I never made it that far, certainly. It results in crazy strong absorption of light at this particular resonance frequency. And if you start to grow the size of the particle, the scattering can be increased like crazy as well. So essentially, when these natural resonances that are inherent for a given particle of a given type of a given size are excited by incoming light, absorption and scattering can be 40 times higher than the same size particle that's made of a polymer, that's not made of these cool metal materials. And because of these new coupled electron movements, you can think of these LSPRs as basically being tiny but extremely powerful electric fields. So you can see that in the kind of gradient and the E field coming out of the particle here on the right. It decays very fast, rapidly away as you move away in space. So they're in the near field of the particle, very, very close, a few nanometer. But they're extremely high electric fields. And I'll tell you sort of what implication that has in a couple slides. I keep calling these resonances. Here's a little animation of a resonance. I think we all kind of know macroscopically what it is. It's when you have some external vibrating system. So that's the hand in this animation. You can force another system to vibrate with a greater amplitude at a specific frequency. And the frequency at which the second body starts vibrating is called the resonant frequency. You can essentially think of the nanoparticles' electron oscillations as being in phase with the incident light frequency. And that's the resonance. So as long as you have that specific wavelength of light shining on the nanomaterial, the electrons will continue to oscillate back and forth and generate crazy high magnetic fields, which can be used for heating or for sensing. And we'll show you how we use that for EVs now. And when you switch off the light, it dampens back to the original flow of electrons randomly, and you don't have this electric field anymore. So the light and the electrons coupling is the key. You have to have both to excite this mode. So of course we could dig much deeper into this, and I won't go through all this math, but it's actually really important to think about the geometry of this light matter interaction. So here's a different way to look at it, with light coming in this h nu here at some angle to the particle surface. And some of the light that couples with the surface at the resonance condition creates these standing waves. And these are actually what the surface plasmons are. They're oscillations in positive, negative, traveling along the surface. They're literally radiating across the surface. And you can sort of mathematically treat the component of the incident light wave as having a vector that's parallel to the surface. So this visualization, the reason why, even though it's a little advanced, why I wanted to show it is because it explains why we have the nano part of Nano-plasmonic. So why can't bulk gold have this kind of surface plasmon? Well, it's actually not possible to generate surface plasmons by direct coupling of light with smooth bulk metal surfaces, because the propagation constant of the surface plasmon is greater than the incident light wave vector. So you actually, there's kind of a general rule that you need to have some metal feature that's about 10 times less than the wavelength of light. And then you can generate these surface plasmons. So these could be small particles that on the whole are smaller, 10, 50, 100 nanometer, or they could be nano-roughened bulk surfaces. So whatever it is on the nanoscale, it has to have tiny nano features, pillars, crevices, spikes, whatever. And you know, back to this idea that it's very geometry dependent. So you have this resonant condition, essentially, which particular colors you have absorbing or scattering. It's highly dependent on the material composition, gold versus silver, for example, the size, the medium in which they're embedded. So you can see there's a lot of work out there of people totally simulating the electromagnetic fields around these different particles. In this case, it's like your eyes are the laser. It's coming from out of the screen and into these particles, and you can see the standing wave generated in the kind of XY plane. I mean, who doesn't want laser eyes? So you can see really that shape plays a big role. The magnitude of the electromagnetic fields are growing as it gets more red from blue. And we'll talk more about hotspots in a minute, but you can see when you bring two particles closer into the bottom, you get extremely high electromagnetic fields. So in general, much of the material science field is dealing with how to really control the size and shape processing to make any kind of shape and size and bring features together, but it's still a big challenge. Okay, so how do we use this for sensing? Well, basically, you can just make simple static measurements of surface plasmons. So the surface plasmon manifests itself as an absorbance. So the particular light frequency that generates the surface plasmon then absorbs all that light. So if you look at the absorbance spectrum of these particles, you can find it very easily. And again, these are just normal UV vises like the plate readers that you have in your lab. And as you see down in the bottom panel here, these are apparent colors that we can see with our eyes. So a lot of these sensing modalities don't need these kind of extra detectors at all. So one of the most common things that you can do, especially powerful for low resource application, is just monitor aggregation. So as silver nanoparticles start to aggregate, you get weird shifting resonance modes. The color changes from red to blue. People, of course, have already started to use this for detecting EVs. So in fact, to determine their purity. So this is an interesting setup. These gold particles, as shown with their kind of typical absorbance spectrum, which means their surface plasmon is in resonance around 520 nanometer, kind of green. Well, if you add EVs, you can make the gold particles cationic. They're just going to non-specifically bind to your anionic. Remember, all EVs are inherently negatively charged. They just kind of aggregate around. And this leads to crazy aggregates, if they were to aggregate, that totally shift the spectrum to weird blue kind of colors, which are shown in the top right. But if there's a bunch of free protein around that is far more numerous than the EVs, they'll instead bind to the gold particles, and they'll form a corona around the gold particle that prevents it from actually binding to the EV. So here's some data. You can see the red and blue sort of visual appearance when the free protein is there or not, and a better quality UV vis curve, the LSPR. And these authors show that this assay was able to detect nanogram levels of free protein amongst EVs just by monitoring the UV vis signal. And so a lot of labs have started to sort of employ this exact sort of assay to measure purity before functional downstream studies. And these authors actually published a 2020 follow-up in Frontiers in Chemistry. That's a protocol, a very detailed protocol paper if you want to try this out yourself. So shifting gears a little bit, probably the most successful and widely known application of plasmonics has been in an instrument called the Surface Plasmon Resonance, SPR. They named the instrument after the phenomena, pretty confusing. And this instrument can detect the binding of an analyte. It's very sensitive thanks to this plasmonic effect. So you can see in the top figure on the left here, light is focused on a kind of 50 nanometer thin metal film through a glass prism. And the subsequent reflection is detected from a PMT. And at a certain angle, the incoming light couples to the metal surface. You get your surface plasmon. So then the light basically all goes away. Now, if a biomass binds to the other side of this, like you can see an antibody-coated bottom side with a microfluidic channel over, the biomass changes, the refractive index changes, and the angle that you absorb changes. So very subtle shifts in angle of absorption, peak absorption, can give you very small biomass increments of binding. And people have used this in cool microfluidic experiments to measure all kinds of kinetic and thermodynamic properties of binding. And a lot of people have started to use this for EVs. So here's one where they were using a panel of tumor-associated markers. So these are different chips. One had HER2, EpCAM, another CD24, another CD44, alongside CD9 and CD63. And they showed differential expression in EVs from various breast cancer cell lines with increasing metastatic potential. There are even more recent applications on top of this, where you can add actually nanoplasmonic particles on top of the EVs after they're on the biosensor, and that increases the refractive index even more, so you have even more sensitive detection of these EVs. In this case, the gold particles aren't doing anything special with light matter interaction. They're just serving as a kind of contrast agent to increase the refractive index. So SPR has excellent sensitivity, typically order of magnitude or two better than ELISA, even without the extra particles added. But they're also really expensive, and there's the devil's in the details when you're doing antibody binding, as I'm sure many of you know, and they're not readily found. For example, we had one at Davis, and it broke and very expensive to fix, and so now we don't have one at Davis. But as you can imagine, some people already started to miniaturize and multiplex this basic technology, making their own sort of miniature SPRs. This one's called the SPRI. And they kind of combine antibody micro arrays with a microfluidic channel. And they also did a sort of investigation into metastatic potential of tumor cell lines. You can see at the bottom right here, their detection of CD9 onto some EVs from some cell lines matched Western blot, quantitative Western blot really well. So, you know, it's exciting, but it's all very preliminary, like most EV stuff, as I'm sure we all recognize, but it's promising. So to really get in the clinic, of course, we're gonna need years of testing still. Okay, so the fun physics doesn't stop with light and planar surfaces at SPR, but probably one of the most exciting new avenues of detection of EVs is using these kind of planar arrays. So this work is kind of been pioneered by Hako Lee's group at Mass General in Boston. And it's based on optical transmission of light through periodic nanoholes that they can synthesize. They call this the Nano-plasmonic exosome sensor, the NPLEX. And it's a type of LSPR measurement where you get enhanced field at the corners in these holes. Remember those simulations, the sharp features actually give you extra electromagnetic field. So in the simulations on the left, these bright yellow areas are electromagnetic field strengths. They tune the hole dimensions and spacing so that this enhanced field is roughly the size of what we know now are sort of the larger small EVs, if that makes sense, so around 100 nanometer. When light passes through, it experiences some enhanced transmission at certain wavelengths based on this interaction of light with the plasmon at these corners. And if the biomass is absorbed in those fields, the effective refractive index changes, you get a slight shift in the peak transmission frequency. So it's a really sensitive biomass detector, but specifically for biomasses of a given size. So it's very creative and cool. And of course, gold is very easy to chemically functionalize. So they functionalized a bunch of these arrays with different affinity ligands towards different surface membrane proteins. Of course, it wouldn't be plasmonics if you didn't spike in some more particles on top. That's what you can see in the panel C at the top here. They added some stars to even increase their shifts even more, be more sensitive to a smaller number of EVs. And they found that when they lysed the EVs off the chip after and assayed the messenger RNA levels, they found them to correlate quite well with the captured protein of interest. Further, they found that they could detect levels of EpCAM and CD24 in EVs from ovarian cancer patients, human patients, seen in C at the bottom here compared to control patients, and even found major differences in patients that responded to chemo or not. So very exciting kind of new approach. Okay, so on to the final part, which is what we can do with these electromagnetic fields. This is my personal favorite. It's what I study in my group, so I'm a little biased. So without getting into too much of the physics, the light scattering that we've been talking about this whole time has been actually referring to elastic scattering. That means that the wavelength of light, the frequency, the color, whatever you want to call it, doesn't change after it scatters off the particle. It just changes direction. In fact, some of the light, a very small portion of it, actually does lose or gain some energy depending on the composition of what it's scattering off of. And this is called inelastic scattering. It's also called Raman scattering. And so the carbon-hydrogen bond, it's vibrating at a certain frequency. Carbon-nitrogen bond has a few different modes or frequencies it can vibrate at. So just like our incident light can couple with the electrons in the gold to pump them in resonance, you can do the same thing with the molecular vibrational modes of different chemicals. So here, the spectrum at the bottom is for a simple phenol molecule. And you can see we have this nice complex spectrum that can be used to identify it. All of the peaks in the spectrum arise from unique vibrational and rotational modes in that molecule. So I'll play this animation here. This is showing it in action. Anyone with, you know, small children and working from home right now knows exactly what this is. You're probably guzzling it every night. That's right, it's our friend ethanol. And here are all of its modes in action. You can see that each one is associated with a particular energy level, but they're all happening at once or near simultaneously. So Raman is this widely used technique to characterize these vibrational modes. But the problem is it's super weak. Only 1 in a million or even 10 million of the incoming photons get inelastically scattered. Fluorescence processes, on the other hand, are 15 orders of magnitude more likely. So Raman hasn't really caught on to be that clinically useful outside of a specific few areas because of how weak it is. But what happens now if you localize this Raman scattered kind of analyte, your EV, your chemical, your ethanol, in between a gap mode plasmon of two gold particles. Well, you get crazy enhancements because of these electric fields. Again, hand wavy quantum physics. And we're not talking like 10 times or 100 times. We're talking like 1 to the 10th or 1 to the 15th power of enhancements, depending on how you can confine the electromagnetic fields to these hotspots. So that means the signal that you observe from a single molecule would be the same as if you had 10 to the 10 of those molecules compared to a single molecule for normal Raman. So it's insane. And this is called surface enhanced Raman spectroscopy, or SIRS. It's a massive field in analytical chemistry and physics, and it's heavily being applied to chemical and biological detection because it's, you know, label free, and this extreme degree of chemical specificity and sensitivity you're getting from both the electromagnetic enhancement and the inherent molecular vibrations. It's used right now in explosive detections at airports worldwide. There's a Raman scope going up on the next Mars rover to detect biomolecules that can indicate the presence of life. Widely used for anti-counterfeiting, analyzing chemical composition of money. Also can be used for art history. That's a really cool field. We can date paintings and archaeologic materials because we know the composition of the substances, let's say the paints that those societies had access to. And of course, it's also being used for biology. So to detect pathogens, small toxins, pollutants, and of course, cancer-related biomolecules for liquid biopsy like EVs. So the basic premise then is to bring your EV close to a nanoplasmonic surface. Keep in mind this phenomena, this crazy magnetic field, electromagnetic field, is a few nanometer, maybe 10, 20 if you're lucky. So you have to bring it very close, and even then you're only seeing a small part of that. More on that in a second when I show you my data. You make the SIRS measurements, you compare, for example, cancer versus healthy. And we know now from many studies in the field that the composition of EVs and their lipids, sterol, small molecule sugars, all these things we can see in Raman are obviously different from cancer versus healthy ones, and even different types of cancers and different stages of cancers. You can functionalize these plasmonic materials with antibody or affinity reagents to capture EVs, and then just do normal SIRS looking at for peaks in general, not necessarily tumor-specific ones, or some combination of that, pull down and then look at the full Raman. Okay, so that's the end of the didactic portion of the talk. I'm going to spend the next 5 to 10 minutes to finish up and tell you a specific kind of case study of how we're using SIRS to look at gold nanoparticles. I mean, at tumor EVs, sorry. Okay, so here's our typical workflow. We isolate EVs, just like all of you. We're still testing what the best method is for SIRS analysis. We see differences. And so we're combining or comparing ultracentrifugation, density gradient, and size exclusion chromatography. And we're typically doing all the normal analysis, NTA and ExoView and TEM and Western blot and so on. I have a new resistive pulse sensing in my lab that's still in a crate, but we're getting that set up pretty soon. So then once we have some level of purified EVs, of course, we would like to not do any purification. We're testing that as well. We deposit them onto SIRS materials. These could be gold nanoparticles deposited on the surface. It could be gold nanopillars. There's a bunch of commercial sources for these, or we can make them ourselves. And then we pop these onto a microscope. And this microscope looks remarkably similar to a confocal fluorescence setup, except that we collect the Raman scattered light on a spectrophotometer and camera afterwards. And we scan around our material to try to image the EVs in this way. So here's one of the typical SIRS substrates that we can buy commercially in the middle, imaged by scanning electron microscopy, SEM. You can see some EVs probably captured on top. And these are kind of rounded gold mounds in a periodic array underneath that. These are sold from a company called Moxtec for about $30 a pop, and you can wash them and reuse them. And we'll sometimes do a simple trick, which is we can functionalize the gold with a thiolated amine. This basically results in the surface being positively charged. So then we can non-specifically recruit the anionic EVs down to stick to the hotspots. And this helped boost our signal a ton. So then we just accumulate spectra over and over and over and over and over, scanning all across, sometimes looking for a particular site, sometimes just randomly, and we compare any samples we can get our hands on, cancer versus healthy, different diseases, different biofluids, isolation methods, blah, blah, blah. And then we do some principal component analysis to dig deeper into the differences between these spectrum. So more recently, we've been exploring this weird SIRS substrate that's homemade. It's very cheap. Well, let's say inexpensive. There's no need for lithography like these previous perfect gold nano mounds or nano holes or whatever. So I do want to acknowledge that this work is carried out by my first grad student, Hannah Koster, and my postdoc Tattoo, who's currently trapped back home in Finland, waiting out this global storm. So rather than using very expensive materials, you actually can just buy a bunch of silica diatoms. These are these little water creatures. You can buy them in powder from Sigma, and they're excellent photonic substrates. So we compress these diatoms. You can just think of them as a source of silica scaffold with a bunch of silver nanoparticles and literally just compress them down to form little strips, substrates. And they end up being porous. You can see in this cartoon in the bottom central here. So we can put EVs in solution on top. They diffuse into the material, get snagged up by these cationically charged silver nanoparticle clusters, and we scan across with our taking our SIRS measurements over and over. So it's important that this kind of porous nature gives us some extra purification ability. Here's a bunch of SEM images where you can see, especially in the bottom middle, how these silver particles get all kind of clustered together to make these super bright hotspots, but they're kind of sparse, so we have to scan around. And in red, you can see probably some EVs, and on the bottom, probably some MVs. The scale bar is a micron here, so those are pretty large on the bottom. So a trick that we use to better interpret the spectra that we get is principal component analysis. So we can rank each individual spectra on 1, 2, or more axes according to the influence of certain spectral features in its own spectrum. So for example, on the left, you can see that each dot represents one SIRS measurement somewhere in the substrate, with red being samples that were isolated from ovarian cancer patient plasma, blue was for endometrial cancer plasma, and white were controls with either benign or no malignancy. And we can rank these according to the main drivers of the chemical differences that we see in the Raman spectrum, and then statistically group them. What we hope is that what will come out is that our samples will split according to type, and that's actually exactly what we're seeing. So we get a clean separation, and so then we can start to say, well, what are these axes representing? What are the chemical features that are actually distinguishing these endometrial cancer from ovarian? You can see up on the y-axis, all the controls are grouping with one of the endometrial cancer. Well, that was actually a stage 1 cancer, a very early stage. All the ones in the under the origin in the y-axis were higher stage. So it seems that we even have some sensitivity to staging. And actually, this is so new, we don't even know what these spectra mean yet. We're still working out the details. But the last kind of point I'll give to you, and this may be sort of more general interest to everyone, is that remember that only a small part of the EV is in this short enhancement region around the plasmonic particle. And we believed that the thick sugar layer found on EVs, which people actually don't talk about too much, this glycocalyx, was actually blocking the signal that could be coming from lipid bilayer and even the intraluminal compartment to some degree. So we used light trypsin treatment to cleave off the sugars, and then we repeated all of these measurements on these clinical samples. And without getting into all the detail again about all this PCA, what we really saw was that all of the sugar-related peaks disappeared from our spectrum after we trypsinized. So we actually trypsinized. And by the way, the size and count of them was similar, so we know we're not just destroying everything. But a lot of the separation that we had previously from cancer versus control or cancer type from other cancer type collapsed. We totally lost it. So we essentially removed probably important diagnostic information from the vesicles by cleaving off all of these extraluminal domains. Okay, so that's kind of interesting. The sugars may be important, and that's just kind of the brief story I wanted to tell you. So I'll sort of wrap up my talk here. I hope you took something away from this presentation, namely that surface plasmons are these special interactions of light with the free conduction electrons of metals that lead to increased absorption and scattering, and then generation of these crazy electric fields close to the particle. And that field is responsible for greatly boosting Raman scattering from biological molecules that are close to the surface, which enable excellent sensitivity and specificity for EVs. So I want to thank my excellent young research group that are pictured below, especially Hannah and Tattoo, all of their data. And then I want to acknowledge our generous funding sources and collaborators, and you can feel free to hit us up on social media. Pictures of my animals and also some science sometimes. And my wife is actually currently about 6 months pregnant. So interesting time to be pregnant here, but if you follow me, I'm sure you'll see lots of cute baby pictures in a few months as well. So last but not least, I'll invite you to check out some of my lab's talks at ISEV in a couple weeks. We have a great presence there covering a lot of plasmonic stuff and then just some plain old ramen too. So for example, one cool thing is we can tell apart lipoprotein from EVs extremely easily with this technique, totally label free. So there's some talks on that. So with that, I thank you for hanging in there for this sort of physics heavy talk, and look forward to any questions or discussions or maybe even some potential collaborations. So thanks a lot.
Thanks so much, Randy. I really enjoyed your talk. And I think we've had several questions that have come in the chat box, although I think that some of the people who are on the call are not able to use their microphones. So I will try to relate the questions to you right now, and we'll see if anybody else has any follow-ups to those questions. So a question comes from Yvonne Couch, and she asks, she wants to emphasize that she is not a physicist, so keep this in mind. And she asks she asks about iron actually to start with. So there are reports of EVs absorbing iron in the circulation. How would that influence or interfere with the signal here? And to what extent does iron as a metal behave like the gold and silver nanoparticles that you told us about?
Yeah, great question. I mean, the short answer is that iron is not tremendously plasmonic, so it's not that useful in terms of what I was showing, but they do scatter like crazy. So they're highly refractive, but they don't have these absorption and kind of colorimetric properties. But of course, then they're magnetic. So there's other options, at least the nanoscale version of iron, that you could actually, a lot of people are engineering these metaparticles that are composed in a single crystal lattice of gold and iron oxide to make magnetic plasmonic materials. So you should be able to see them scattering. In fact, I've thought for a long time that these highly refractive particles that we often see on NTA or flow are probably some bioaccumulated crystals or maybe even metals. And we're looking into that ourselves. But in general, they wouldn't have the same sort of utility.
All right, so the next question is about what you could possibly do downstream. So you're using these gold labels. Could this labeling somehow be coupled with sorting? And if you label and sort, can you then remove the label so that you could possibly use these EVs in functional assays?
Yeah, we're trying that exactly as I said. That's very perceptive. In fact, that's what Josh and Jennifer were trying to do originally, were to use these as sorting labels for flow, so that you have this highly refractive now group. So you're not even using the fluorescence on flow. You're literally just using the side scattering and forward scattering and making sorting decisions based on the refractive index. That's excellent promise. And now, if you can think about combining these with the magnetic properties of nanoparticles too, you can even think of really crazy modalities where you're doing cool microfluidic separation. And as far as removing them, they're biofunctionalized literally the same as any other antibody aptamer peptide. So if you would have to engineer some sort of, esterase reactive cleavable linker or dithiol or remove them with some sort of, like removing biotin and streptavidin is tricky. You need some salts and acids, but it's possible to do that later. Good, actually, yeah, just having them there, their weight might be helpful to change the density or where they go in a density gradient or something like that.
True. Good, yeah, and it looks like Lilia had a similar question, so I'm glad that we could address that. Maya, you're up next. I'm going to unmute you here.
Hello, Randy. Thank you for the fascinating talk. This is really cool stuff, and I see great potential in it. I'm interested, since I have some background in glycobiology, what's the deal with the glycans? Are they, because you said that they mask the lipid detection. So having in mind that vesicles have all kinds of different molecules in them, why are glycans interfering with
yeah, so the interference from the glycans for us are strictly based on the size. So the coupling with these gold particles to get this boost is size dependent. We have to be within a few nanometers. the sugars just physically get in the way, basically.
Yeah.
And so we cleave them just to then bring the lipids closer, and then we lost a lot of our sensitivity. So probably the sugar composition, and I know a lot of people, probably even you, are starting to find that these sugars can be really diagnostically important, functionally important. So yeah.
So does it mean that you actually cannot detect what's inside vesicle if the vesicle is big?
Yes, exactly. We might, so if we have like a 10 nanometer kind of depth, and it's not linear, it's decreasing it. So maybe we can see 20 nanometers, but 10 times less sensitivity. The glycocalyx
on the other side,
yeah, so we can see a little bit of, so sometimes we see some nucleic acid, sometimes we don't. Yeah, we were trying to see, can we see these, you know, fabled DNA, which can be on the outside of EVs sometimes. We have some evidence that might be the case. Yeah, so very distance dependent. The normal Raman without plasmonics, anything that's in the focal volume can be detected, so we're not limited by that. So when we use regular Raman, we can kind of see through the EV, but with the SIRS, it's only, it's spatially dependent.
Thanks very much, and thank you for clarifying some physics stuff for us. Thank you.
Thanks for the question, Maya. Next up is Mike Pilatus. Mike, just unmuted you here.
No? Okay, Kat.
There we go.
Can you hear me now? Okay.
Yes.
Thanks, Randy. Very nice talk. My question is on your Raman spectroscopy technique and the sensitivity to EV sample purity. And also, do you see, you talked about doing several different separation techniques. Do you see difference in the spectroscopy depending on the technique you use for EV separations?
Yeah, good question. We sure do. So two of my students' talks at ISET will dig specifically into this, combining different modes of isolation and then using them in series, SEC followed by density gradient or vice versa, for example. What we mostly see are differences in the extent of lipoprotein when we're going from clinical samples. So of course, some modes are based on size, they're taking out larger lipoproteins. Some are based on density, they're taking out different density lipoproteins. But lipoproteins aren't all the same. They have a range of sizes and densities, just like EVs. So you need orthogonal isolation techniques to perfectly purify them away, and we can see that readily. So we have lots of experiments that my students will talk about more in detail that are spiking in purified lipoprotein that we can buy to sort of measure to what extent we can see them. But even in vitro sources where we don't have any lipoprotein, we see major differences in the spectral composition of EVs according to their isolation methods. However, when we globally look at different cancers, different patients, and then each one of those samples isolated differentially, the differences amongst patients is much greater than the differences amongst isolation techniques. So it seems like it's not as critical. We can still get diagnostic information not depending on what isolation method we're using.
Interesting. Well, thank you, Mike. And I'm going to unmute Martine. Martine, you had a question as well about sugars.
Hello, Kenneth.
Hi.
Nice to see you again. So the question is, since you find that carbohydrates are more willing to interact at the initiation of the particle, are this sugar more expected to be more useful for detection in the cells or different than proteins?
Yeah, I mean, it's a really good question, and we don't know. We were really surprised. A lot of people are doing these light trypsinization or other type of experiments to cleave the sugars, and we thought certainly we would get a better diagnostic sensitivity. And we only have just now recently realized, like literally in the past, like in March, like right before we all had to shut down, that the sugars were so critical in differentiating the different types of cancers. And so we don't really know what to do with that. I think one option is I know some companies are making antibodies and affinity agents for sugars in particular. So maybe we can think about pre-isolating EVs depending on their sugar composition. I'm actually definitely going to be exploring all the talks at ISEV that are dealing with glycans to try to learn more about this and see kind of what people are doing to see how we could sort of help with the ramen story. But they seem really important. And yeah, they're sort of epitopes, or the specificity to the degree that it's there could be a really cool handle to increase the specificity of diagnostics. Or even for engineered therapeutics, who knows what the function of these actually are.
Thanks, Martine, for that question. Let's see here. Lilia, you have a question here as well. Let me unmute you. Go ahead.
Yeah, thank you for the talk. My question is about the agglomeration, whether it's possible to say that, okay, now we're talking about the large vesicle, and that's the case that there is only one particle count, or it's basically just agglomerate of the different lipoproteins or the small vesicles together. If there is any way to see that on the spectrum by your method?
That's a great question, and the short answer is we don't know. Whenever we see these signals, we're not sure that it's arising from a single particle or from an aggregated clump. It's a great point, and I think the same issues that we have with any single particle technique that we're looking at, it's very difficult to know if it's really one thing or an aggregated clump. I think there's probably, you can direct agglomeration pretty easily with salts and things. So that might be an interesting line of work to start to do that on purpose to see if we can see some sort of spectral differences. But it's something that we don't control for in these studies. Definitely kind of an unknown for us.
Okay, all right, thanks.
Yeah, so Maya Kosanovic has also just sent me a message saying that, just reminding us that we can isolate different sugar-containing EVs with lectins. So that's one.
That's right. Okay, cool.
We can do.
Yeah, that would be really interesting to try that. And of course, the lectins would have their own Raman signal, so we can tell extent of contamination and all these things.
So we're getting towards the end of the hour now, and I want to maybe conclude with a question I think that's on everybody's mind. And that is about money. So you mentioned that some of these instruments are quite expensive. So what are we talking about in terms of US dollars or euros?
Yeah, so the confocal Raman setup that we built in our lab is about 150K. I think a commercial one would run about double that. However, there's also, and that's absolutely what you're going to need for spontaneous Raman. So to get the sensitivity for this really weak spontaneous Raman, no plasmonics, you need very advanced instrumentation. And there are examples in the world, especially in Germany, where some of the Raman experts really are working, where they have full-scale Raman scopes that are $500,000 in a clinical workflow to detect pathogens and bacteria. So it's not too unfeasible. But there's a totally other side of this, which are handheld little spectrometers, even using your smartphone, which when you have the sensitivity by these gold nanoparticles that boost it 10 to the 15 times, they all of a sudden become really viable. So these detectors can be $5,000 to $10,000. And trust me, a bunch of people are working on to try to scale those numbers down right now. And then you could detect them very simply. And the lithographically produced SIRS substrates, like the mounds I showed you, are expensive, $30 to $50 per substrate. Of course, if they're used in a diagnostic scale in the world, they would drive the price down, but they can still be considered expensive. But making gold particles is extremely cheap. So we're talking a couple cents for way more than enough to characterize hundreds of patient samples. But they're just not there yet to be, to be diagnostically applied. But you can imagine, like with a really sensitive SIRS setup, that you could keep it under 20K for a whole kind of clinical workflow.
So it looks like we're moving in the direction of having these technologies available to a wider,
exactly,
a wider population, a wider set of applications.
Yeah, which of course will improve everything, the engineering of all these. Yeah.
Great. All right. Well, that will wrap up our session for today. And Randy, thank you so much for joining us.
Yeah, this was great.
Thanks everybody for your attendance and for your great questions. Randy, we'll look forward to seeing some of those baby pics coming up. And I'll also look forward to seeing many of you, hopefully all of you, at the ISEV meeting coming up in a couple of weeks. And as Randy mentioned, there will be some presentations of his work there. I know that I think you're one of the more prolific contributors to the meeting, so that's great. Thank you so much for supporting ISEV. And if anybody would like to join the meeting and hasn't registered yet, you can just go to isev.org and get your registration. You'll be able to access everything for 2 months after the meeting as well. So it's a nice feature of this meeting that we haven't had before. So thank you, everybody. I hope you all stay safe and healthy, and we'll look forward to seeing you again soon. Take care, everyone.